Today, so that's the a turning point for this Q&A. I would like really to go back a year or maybe 14 months back and explain you what we did last year and where we want to go for next year and maybe the year after. So that's from foundations to integration. That's a very fancy
Okay.
start. So I'm going during this Q&A today. I'm going to bring you in on a trip. We are going to travel the whole world. This is the map where you have the Myhre syndrome scientific research sites. And you see that it goes from South Korea to Australia to the Pacific coast of the US, several sites in US, but also a lot of sites in Europe, in UK, France, Belgium, and Spain. So we are going to go through all the different project and program we have, and every time I will tell you where that project happens. So in 2025, what we did is really focus on how can we understand, start to understand, and how can we test. So the first thing was to build the tools to have the best idea for discovery. So what we did is that we established a preclinical platform, and I'm going to recapitulate what we did, give you some results. And we developed the patient-derived cellular models, and we initiated the humanized animal model in that is almost done. We have the pups. Um, and then I will explain you where we want to go in 2026. And the idea is really breaking silos. That's the 2026 spirit. The so starting from the beginning, Myhre syndrome results from a single base change in the 3.2 billion pairs of the human genome. Yet, even with one very tiny change, we have a constellation of manifestation from neurodevelopment to decreased mobility, stenosis, respiratory, cardiac complication, hearing impairment, or pain, or even other short stature, and everything will depend on on patient from patient to patients. There is a real heterogeneity, and it's really multi-systemic. So where do we start? We want to understand, and we want to test disease. Where do we look, and what do we use to start these studies? So usually what we do in science is that we take models because we cannot um, go directly on human. So the modeling is really a challenge, especially in Myhre syndrome, because it's difficult to biopsy every organ in a patient in general, but with Myhre patients, biopsies are really not recommended because of the fibrosis and scarring that can be an issue. The animal physiology does not fully replicate human variability. If we were mice, we would be cured of every disease on the planet. We are human, so the mice model is not really ideal either. If you go then to individual cell types, that's great, but you only capture one fragment of the disease. So there is no perfect model. But what we did is that we built a platform starting with non-animal models. So starting as the patient as the cornerstone and not starting from the animals. We start from the patient. And what we did is that we started with the blood of the patient. So we took blood from patients with the I500V mutation and patient with the mutation R496C. We took also sibling controls. So they are same sex, same age. They have this similar genetic backgrounds. We did not engineer the cells that had mutations. So that's what scientific called isogenic. It means that as a control, you just rectify the mutation and you take that as control. The problem when you do that is that, okay, you are going to compare exactly the same cell, but the problem is that you might trigger other modification in the cell. So one of our strengths is that we have different type of control, and the good one is the sibling controls. So we took those cells from the blood. We just took blood from the patient. Then we isolated the cells and we transformed those cells back into a kind of embryonic cells. It's that what we call stem cells. So they are not coming from embryons. They are coming from the blood from the patient, even adults patients' blood. But then you take the cells and you bring them back to the stage similar to embryonic. So when you were even before embryonic, when you were just a mass of cells, all the cells the same, and from there, your cells, when you grow, are going to become muscles, brain, bones, whatever. So we took those cells from the patient, put them back at the stem cell level, and then from those stem cells, we can reprogram them in whatever we want. If we want to have brain cells, we can. If we want to have vascular cells, and we chose endothelial cells, we can. If we want to have muscle cells, we can. And those are cells from the patient. But we didn't have to go and take a piece of muscle, a piece of the skin, or a piece of the brain, which is not never done. Uh, so what we did is that we chose for now two type of cells to reprogram into the endothelial cells, which are part of the vascular system, because Myhre syndrome has a lot to do with fibrosis and the vascular system and cardiovascular system. We have microglial cells that are brain cells that participate in neuron death and death and survival regulation, and also forming or killing neurons connections, what we call synapse. So those are the cells that we have now. What's for 2026, we are going to go on with differentiating new cells such as neuron specific muscle cells that are around the vascular system, the smooth muscle cells, cardiac cells. Well, we can go astrocytes. We can go in different directions, but we are going to expand that. What we did with those two that we have already since that we developed in 2025 is that we characterize them and we try to identify what's the difference in their function, in the way they function compared to the sibling. So the mutated compared to the control. And what we saw is that the microglial function, so the cells that we have, the brain cells, in the patients function 50 to 60% less than the control. For the endothelial cells, we checked, we do an experiment that is we trigger a wound. So we do a scratch on the cells, and then we look how the wound is going to heal. So the migration of the cells and how it's going to close the wounds. That's what we call wound response. And here we see that the patients have 30 to 50% more. So the one number is not that there is a variation, is that there is a difference between the two variants. So you have for microglial function, you have one variant at around 50% and the other variant around 60% of the decrease in functioning. And for the endothelial migration and wound response, for one variant, you have 30% increase, and for the other variant, you have 50% increase in the wound healing process. Those were really, it took time to establish the assay and everything, and those now are standardized and good to go. The next step, besides creating new type of cells from the patient cells, is going to build what we call microphysiological system previously named organoids. I think they didn't like the name, so they make it more difficult for French people. Now it's microphysiological system. Um, that's um, that's a way to really capture cell-to-cell communication because in your body, you don't have only endothelial cells or only microglial cells. You have for microglia in the brain, for example, you have the astrocytes, the neuron, the microglia, and they all communicate together. They send signal one to another. So it's great to have the easy, fast, and standardized assay, so measurements of the difference with the microglial cells. But if we want to go to a more complex and more relevant for the disease model, then microphysiological systems are even better. We can do multi-system with, for example, neurons, astrocytes, and microglia, and look how they communicate and how we can modify that, and the difference between those systems in the patient and in the control. It the what we used to call organoids reflects more the organ level complexity. So that's something we really want to do. Um,
Ahmed,
yeah.
and on the variants and the findings, is it one variant that tracks the lower end of both sides, or is it does the variant change? So is it the 500V that's lower on function for microglial and endothelial, or does it change around?
Now it changed around.
It
So
changes around.
yeah,
So one's lower on microglial but higher on endothelial.
yeah.
Right. Okay.
Yeah, but to confirm that, so that's just an indication that I give you, but to confirm that we need to have, because that's just one N of one for the 500 patient and N of one for the 496 patient. So really to confirm the difference between the two, when we talk about patient cells, we need to have different patients
Hmm.
so that we can confirm that in 10 different patients with the 496C, the microglial is going to be the function is going to be less than in 10 patient of 500. Now it's just a one and one. So we need to be careful on those type of conclusions. But that's
Were
from
you
what
expecting that to see that there was like it would almost move around what the higher response was?
well, there is an indication that maybe there is a difference between the variants, but
Hmm.
it has never, in studying the patients, we can see that there is a tendency, for example, for hearing loss, definitely difference between the two variants, but it's not really established with the mutated SMAD.
Okay.
So that's a way to go. That's an indication. That's the a progress in characterizing the two variants. Um, so with this essay now, what do they allow? What did they give us? Progress on the disease understanding, and what's really important, I mean, progress on the understanding is important, but the other thing is that we have a quantifiable therapeutic testing, and that's really good. It means that now we can test drugs in patient cells. So this work was done at IXLs in California and at New Site Lab in California also. So to go more in details, so now you can see here that you have so that's the microglia. So you see that the patient's microglia really functions less than the control. So you have that window here. And for the 496, the same, the microglia function less than the control. So we are going to use that type of assay, I mean that assay to screen drugs. This is the assay for the brain, but we have also an assay for the vascular endothelial cells. And what we want to do is that we want to put drugs on the cells and see if we can bring that curve up to the control level. So in
Mhm.
fact, to remove that difference. And that's the way we are going to screen drugs. In that model, the drug is screened in cellular model, in organoids, and then in mice, and then in human. So
I
what
mean,
we achieve, yeah.
you mean drugs that's all already in the market? That's what you mean?
Drugs that are in clinical trials now,
Hmm.
and also from Spain, new drugs.
Hmm.
So we are going to also include drugs that are on the market, like the pirfenidone, for example, or this kind of thing, just to see as a control and go on with the characterization. But the most interesting drug for us are not yet on the market. They are in clinical trial phase two, one in phase three. So we are going to test different uh, advancements of drug from new chemical to and clinical trial or phase two, phase one clinical trial.
Okay.
So the advantage of the platform is that it's derived directly from the patient. So the first test we do is really on the patient. It's not on an animal and then you go in human, and if you're lucky, it works also in human. It starts from the patient. It's stable and really reproducible, the assay we chose. It's not that we chose the assay to reflect exactly the disease. What we wanted is to have an assay that is stable, reproducible, meaning that if you test a drug in January and then you retest it in March, you are going, or in a year later, you are going to get the same results and you are going to get the cells that react the same way. So we have a functional readout. It means that it's really how the cell function. It's not just in a tube, and very reproducible. And we have a strong signal versus control. So you can really read if there is a modification. So it's a dual use. You can test potential treatments in expected disease pathways such as TGF beta or unexpected mechanisms. And you can also use that to explore disease mechanisms directly in the patient-derived cells. So I'm going to stop here and ask your reaction. Do you understand? Is there things that are really not really clear for you, or things you really don't understand? So please just tell me. Should we go on? Do you have any questions?
Um, so it's actually only from two patients, these cells?
Yes. So there is one patient with of each.
Variant.
Yeah. Yeah, because
So,
the
if
cost,
we
yes.
collect more all around the world, what you will do more from these kinds of cells? That's the idea of the collection of the blood now?
Well, the just you up from just what we said. So if I can go back.
Mhm.
No, sorry, tack. So here, from step one to step two, that cost us almost $100,000.
Okay.
So if you want to repeat that with two or three times, you see the amount of money we are going
Mhm.
to spend.
Mhm.
So we start first with one patient. That's usually what everybody does. So you start with one patient, and if you are a multi-million foundation, you can increase your bank with different patients, but it's very expensive.
Okay. And if we found some reaction which are good for example, some drugs, uh, it's not meaning that in the real life, for example, it's only on special cells, on the cells, but the effect all around the body, we cannot know, we cannot, you need to do all the process and tests, yes?
Yeah, so that's just the beginning.
Hmm.
Now we are going to So I told you that in 2026 we want to develop the organoids, so more complex. So the drug will go from, so you test hundreds of drugs at the cellular level, what I just showed you in here. So here
Mm-hmm.
you can test 100, 200 drugs in 2 weeks. Okay,
Okay.
so that's what we call a bigger throughput. Then you go to more complex, like the organoids, and you can test 20. Then you
Mm-hmm.
go to the mouse model and you test one, and one is going to cost something like at least $10,000.
Mm-hmm.
So the more you go, the higher you go in complexity, the more it costs, and you have a better understanding on the action of the drug, but at the same time, it takes way more time and way more money. So you cannot do 10 drugs in a mouse. You don't do that. You select one, maybe two, and then you go in the mouse. But at the beginning, when you have a lot of different drugs, then you start from something that is might not be recapitulating the whole disease, but at least you can test a lot of compounds and make
Okay.
a first selection. So that's the reason why when you want to do drug discovery, and especially we chose to have a platform. So it starts with the cells, and then you have the organoids, and then you have the animal model. Um, and then it's, well, so that's, there are no ideal models. You need to have all of them, and so that you can have a full pictures of and different level of testing for the different treatments. So that's the, why do we have that? So after the organoids, the microphysiological system, there is, unfortunately, you have to go to animal models. You need to have an idea about the drug or the treatments in a whole organism to validate it. You need to know the distribution of the drug. So if you give the drug, is it going to go to the brain? Is it going to stay in the liver? So you need to understand that. You need to understand the toxicity in the whole body, and you need also, you check also the efficacy. What is the dosage, and a whole type of criteria that you are going to study. The ideal to go in a whole organism before going to human would be to go in a non-human primate. The issue is that it's very costly. It takes more space than mouse. The breeding, the reproduction is the breeding of the animals is way lower than mice, and the amount of drug that you have to put in the animal is way bigger in a non-human primate than in a mouse. So usually people, I mean, scientists go with mouse model because they breed very easily. They are very small, and they are easy to manipulate genetically. But the cost is really, you reduce tremendously the cost. So that's the reason why most people go for mouse model. So what do we have in Myhre syndrome? There are existing models with disease-relevant manifestation. There is the I500V mouse model of the Professor Valérie Cormier there at the Institut Imagine in Paris. And there is the inducible, so they are slightly different, I500V mouse model from Mark Lindsay at MGH in Boston. So what was missing, we do it at the foundation. We created the R496C mouse model at Ausgene in Australia. We selected that company because they have a technology that is very innovative and that will allow us uh, to be easier in the gene editing evaluation and testing that we want to do in our program. So again, there is a dual use of the animal model. There you can test the same potential treatment in expected disease pathway or unexpected mechanism, and you are going to explore disease mechanism in a whole organism, even if it's not human or primate. So now I'll talk about the tools, what we need to understand and study and test. Okay, but now talking about treatments, because that's what we all want. We want treatments. So talking about treatment, where do we start? Well, we know already a few things about Myhre. We know that it's a mutation that it's a missense mutation, so meaning that there is only one base pair that is mutated on only one gene. So the first thing we thought was gene editing. So the target is really to correct the mutation directly in the gene, in the DNA. We tried already in 2025 a certain technique that worked more or less for the 500 and didn't work for the 496 variant. So we changed, and now we are partnering with Revity in UK to work on the base editing on both variants. For the other thing that we know about Myhre is that there is a protein gain of function with the mutated SMAD4 binding excessively to SMAD2/3. So that's something that we know that was demonstrated by the group of Maria Macias in Spain, by Suleimo in Georgia in the US, and by Hongmei in Harvard. So we know that. So what we can do is already act on that. So Maria Macias in Barcelona in Spain did high throughput screening on 260 molecules, and she identified over 100 compounds. So the strategy was to produce the protein, the mutant SMAD4, in a tube, if you want, the SMAD2 protein, put them together. We know that they are going to bind or bind excessively if there is the mutant. And you add different molecules on it, and you see which one is going to separate them or destabilize the complex. So that's what she did. And she ended up with over 100 different molecules. So those molecules, okay, that was in a tube. We know that if you take the protein or even a part of the protein, it's going to act on that binding together, which is the issue with Myhre. Okay, but if you take the same molecules, you put them in a whole cell from the patient in contact with thousands other protein moving really fast in the cells and all the function of the cell, is it still going to be efficient? So that's going from a tube to a cell. And that's the project of Maria Macias is to send those compounds to California, to new sites where we have our preclinical platform, the microglial and endothelial cells, and put those compounds on the cells and see if we can bring the activity back towards the control level. Okay, so that's the next step for this one. And from those, we are going to calculate the curve where it's the most efficient for what's the concentration of the compound that is the most efficient. She's testing also the toxicity of the compounds, and from there we'll go to the next level and slowly go up towards human. What we know, yeah.
Do you have any idea on how long it will take to get through the 100 compounds with the model?
With the cellular level, at the cellular
Yes.
level?
Yeah, through new site.
Uh, First screening a month maybe, and then the dose response another month or two.
Okay, so relatively quick.
Yeah, that's the whole goal of the what the whole thing of last year was really to select the right assays in the cells so that it can go quick.
Okay.
So we spent a year developing that, but now that it's developed, we can really screen compounds on it. So the next things we know is the manifestation. And one of the main manifestation is fibrosis that triggers a lot of different. I mean, fibrosis is not really a manifestation. It's a mechanism of the disease that triggers a lot of issues in the patient. So what we decided is to target fibrosis. And for that, we chose to fund a project in partnership between Isabelle Richard at the Geneton in Paris and Professor Cormier at the Institute Imagine, also in Paris. That's easy, they are close. And they are developing a treatment based on CAR-T immunotherapy, which is used largely used in cancer patient now. There's over 1 million patients that use that technology. So we know that the limited toxicity, the limited side effects, and we use that technology, modify it so that it's not going to attack cancer cells, but it's going to attack fibrotic tissue. So the team of Isabelle Richard is more in developing the technology, the Fab CAR-T, and the team of Valérie Cormier there is she has a mouse model, and we are going to send her also the 496, and she's focusing on characterizing the fibrotic tissue in the two mouse model, and she's going to test the Fab CAR-T with that the Geneton is developing now. So I have a video from Mathilde Magro, who's working at the Geneton for us. So she produced in a very standard way, and she established everything. I'm making it bigger. So she produced the T cells that are able to localize and identify the fibrotic tissue that express the FAP, and they are going to attack those and not the others and kill them. So that's here you see the white, bright white are the T cells that are modified to attack fibrotic tissue, and the long cells are activated fibroblasts with the FAB. And you will see that the T cells, they touch the cell, and then that's the role of a T cell, and kill the cell. And in less than 20 hours, they killed all the cells that were in the well. So you see how you see that one already disappeared. One that was, I don't know if you see my cursor. Well, so one already disappeared, and then you see the others are going to lose their shape and slowly become a kind of big bag and die. So you see how the small white T cells are attacking the other cells and killing them. So if you wait just a few more seconds, you will see that they all disappear. And that's it, and that's the ends of the fibrotic tissue. I'm going to go back to come on.
Hey, Armella, I should know this, but is it a one-time treatment or do you have to do it periodically?
So you will need to do it periodically.
Okay, that's what I thought. Yeah.
Yeah. But this treatment wouldn't be a chronic treatment that you give to kids until for all their life. That's a treatment when you have a fibrotic event that is really damaging for the patient. If you
Dangerous.
yeah, it's really in particular case. It's not just to improve the mobility of your joint, for example. If you see that there is fibrosis in the liver, fibrosis in the tracheal stenosis, then you use this kind of technology from the beginning. When you see that there is this fibrosis that is building up, you do this treatment until the fibrosis is down. And maybe 2 years later, if there is the fibrotic event again going up, then you can do another treatment. But that's definitely not to use all your life. That's not for chronic. So that was in the mice cells. We sent the patient cells, patient blood cells to Mathilde a week ago, and now she's going to go from the mouse to the patient. So we'll see in about two months where she is with the 2 or 3 months, because she needs to grow the cells.
Armel, that
Yeah,
looked like amazingly effective. 24 hours, get rid of the fibrosis cells. Absolutely amazing.
less than 24 hours.
Sorry, less, even better, even better. Less than 24 hours. Is that what would have been expected? Is that just in terms of kind of normal response times for using
So
that kind of technology? So how it look? I mean, for us, it looks amazing. But is it a good amazing, or is that a normal amazing, or is it even slow?
it's it's good. It's good
Good.
results, because usually it's a bit less fast, but that might be, I don't know, because of the way she isolated the T cells. But what's really important is that she can repeat the experiment, and have the same quality of the T cells modified. The CAR T cells are really standardized. So she knows now how to modify the T cells and in a standard way, and have really robust reproducibility and efficiency, and a really nice efficiency, higher than the one she compared to with the another technology that was used before. So what the technology she developed is really efficient. That's really good.
With the Fab CAR T, can you activate and deactivate it? Because I guess the fibrosis cells there, obviously excess fibrosis is a problem, but fibrosis is also needed for kind of healing and regeneration. So how do you control the extent that the CAR T,
Well,
the cells act?
the CAR T they are not living forever in your body first.
Okay.
And what she did is that they have, in fact, the FAP, and they are going to develop another. In fact, the T cells is going around and recognize something on the other cell that she's going to kill. So there is this FAP on the fibrotic tissue. But what they are doing now is that they are analyzing the patient cells and try to identify another one in the fibrotic, only for Myhre. I mean, only maybe in other patients, but in Myhre patients, in those tissue, identify another marker tag, if you want, on those tissue, so that the CAR T are going to attack only those.
Okay.
And what she did also as an experiment, she took fibroblasts, normal fibroblasts that are not in fibrotic state, and the CAR T she has developed do not attack them.
Right. Okay.
They
Okay.
do not kill them. So we have the safety that those Fab CAR T should not attack the non-super fibrotic tissue.
Thanks, Anna.
So that's what we did last year, really starting from what we know, initiating treatments or program tackling the things we know. But there are a lot in Myhre that we don't know. And particularly, we know that the SMAD4 mutated is going to bind more to the SMAD2/3. But how does it behave with all the other thousands molecules and proteins in a cell? We don't know that. So what we did is that we offered the opportunity to Sule Mo. He's a fantastic scientist at Emory University in Georgia. And what he did is that he studied how the two mutants, the I500V and the R496, protein are going to interact with about hundreds of other proteins. So what he did is that he produced the mutant protein, he put that in a tube, he not in a tube, in a kind of other cell. Sorry, it's not in a tube. Um, he produced hundreds of other proteins that are known to be important in fibrosis, in neurodevelopment, in different type of in the production of extracellular matrix. So he went to produce hundreds of proteins, and then he put them together with the mutants, and he compared the way they bind together, each of those hundreds proteins, with the wild type SMAD4, so with the control. And what he found is that it's not only SMAD2/3 that is altered. There are other, in fact, transcription factor for the scientific people here, other proteins that interact with SMAD4 mutated not the right way. And what he saw that it's also variant specific. So there is one of those protein that is more or less common to both of the variant, but there is one variant that is really where it binds really more, the 496. And there are other protein that binds more to the mutants in the 500. So he sees really a difference between the two variants. So all those new protein that we didn't know were not interacting properly with the SMAD4 now are potential new therapeutic targets. So the next step is to validate that those discovery, those new targets in the patient cells. So now we are going to look at that, how those 4 or 5 different new target that we have are behaving with the mutant in the patient cells. And not only in the microglia and endothelial cells, but in really different type of cells to be sure, from the patient still. So to really be sure that those targets can be common to different manifestation. So we are going to check that. And the other way that might be a bit faster is that there are existing molecules for those new potential targets. And I'm going to order them next week and send them to new sites in California to put on our endothelial cells and microglial cells and see how the patient cells react. So that's also something else to go further in those new target validation. Do you have any questions?
Just confirming my understanding more than a question, Amel. So this technique has shown that there's other proteins that the SMAD4 interacts with, the mutant SMAD4 interacts with. So by knowing that, you've then been able to identify other pathways or potential other pathways from a treatment perspective. And some of those kind of new proteins that have been found have already got drugs available or potential drugs available that interact with them. So that's what you're going to take those drugs and put them back through the Myhre testing that you've already done.
Yep, exactly. You
Okay,
understood
thank you.
properly. So it's new opportunity of treatments.
Yep, brilliant.
Yeah, that the tricky thing is going to convince the pharma who have those molecules to allow us to.
That's a
But
licensing issue then,
we can
presumably.
test them. That's no problem because it's research. But then after, but let's first test them and see
Is
what
that
happens.
a licensing issue, Marmel? So it's the license for certain things, and that you'd need to pay for licenses to buy that, to be able to use them for
Well,
something
you need
else.
to convince the people who own the molecule that they should try it with Myhre patients, and that's not an easy thing to do. So, but that's
Yeah.
another story. First, we need to check if they work. That's the first step. Go through the preclinical platform with those molecules and see if we get a result or not. So what we want to do with this platform, and I think it's really important, is to not to use the traditional model that goes from preclinical to clinical. So you go to cells that you modify, and then you go to the mouse, and then you go to human, and it takes forever. And we are some of the treatment that goes along fail, and um, we try to get a new approach. So our approach, which is not conventional, I call it a bidirectional validation loop. So that's a very fancy way to say just that we select drug that we think, and at the same time we use them in compassionate use in a patient, because compassionate use is only for one patient. And those results that have to be communicated, we use them to inform the platform and go in selecting the right new models, the right assays. So it's really, instead of going from the cells and the animals to the human, we do everything at the same time, and we feed the system so that we can really improve the engine of drug discovery. So that's not conventional, but the opportunity we have is that we are going to start compassionate use with one compound that we think is going to be very efficient. And this is going to be used to validate the fact that if the compound works also in the cells, it proves that it's a good model, that it's a good way of testing, that it's good assays. And at the same time, we have so many different manifestations that we can also, thanks to that, looking at the patient, see where we can measure in the patients in a very efficient way how the drug works, and if, not how it works, but if it works, and the efficiency. So that's really a kind of loop feeding, and we have to align the outcome measures, so measure approximately the same things in patients, in organoid models, in cellular model, and in mouse. Not exactly the same, but on the same level. For example, we have fibrosis. Do we go for a skin organoid to look at the fibrosis? Do we go for a vascular organoid? Which type of vascular organoid? Do we go for a cardiac organoid? So having the information integrated from the patients and everything, we can also decide in a better way what organoid we want to develop, what next cell type we want to develop. So that's a dynamic that we have, and I hope it will work.
Amel, why isn't that the typical approach? It sounds eminently sensible to do it like that. Why isn't it normally done like that?
Because there are very few compassionate use allowed
Yeah.
from the companies, not from the FDA, from the companies. It's difficult to convince the companies, and I don't know. It's just, for me, it was obvious, but I don't know. Other people don't do it. Maybe because they
Is there
don't
an
have.
ethical fear, or why are the
No,
companies reluctant?
I think it's more a question than the compassionate use is performed by clinicians, and the platform preclinical models are more made by biologists and scientists, and there is this kind of break between the two, or even the screening is done by chemists, and the chemists don't communicate with the clinicians. There is this kind of blocks, and maybe it's because they don't have the same training, they don't have the same expertise, so they have issue understanding each other's, and it's not a something common to have really the whole, from chemists to clinicians, to be in the same room and talk together. So that, I don't know, maybe that's the reason why it was not done, but I think it's also because um, it's not every rare disease that has the opportunity to have compassionate use.
Okay.
So That might be one of the reasons. So if we are getting there and with what we developed last year and what we are going to develop this year, the urgent thing to do is really to define the trial designs and the endpoint. Be ready for clinical trials. We really need to do that. And the thing is that the clinical trial design is going to be different if you do a FAP CAR-T treatment or a gene editing treatment or a small molecule treatment. But the endpoints shouldn't be that different, but just the design might be a bit different. So we need to identify endpoints. Endpoints is things that you are going to measure in the patient when you try a treatment. That's what we call endpoints. So how can we decide what we are going to measure in the patient to see if a treatment is efficient and safe, and how we are going to measure it? So what we know from now, what's the knowledge we have now? There is one ongoing clinical trial on Myhre patient in France with the losartan. So they have specific endpoints. There is the trial is designed, but it's excluding all neurological endpoints. They have only endpoints that are related to bones growth, joint mobility, cardiac, respiratory, but nothing on neurological endpoints. And I would like to include this part in clinical trials. We have this compassionate use that we want to get, that it's hopefully going to get approved. That will imply systematic reporting to the FDA about all the potential side effects and the benefits. So we are going to have a mine of information on how to monitor a patient. And we have growing data collection all over the world with more and more patients that are identified, so more and more clinicians that get experience and qualified for Myhre syndrome patients. We have at least 3 multidisciplinary clinics, and I think if we go around more, to my knowledge, 3 multidisciplinary clinics, it might be more. We have 2 in the US and 1 in Europe, and we have 3 Center of Excellence, 1 in the US and 2 in Europe. So we start to have quite a few groups everywhere in the whole world that really get experts in Myhre syndrome patients.
And how do you connect between of them? It's actually by you. You are the coordinator between the groups.
We have working groups. So now, for example, we are doing. I will talk about that later. I'm working with all the different clinicians everywhere, and we have working groups and studies all together.
Okay.
So to prepare for the clinical trials, we need to build evidence. We need to do an observational study for at least 6 months. So we are developing a prospective observational study. So starting from a certain point and go further, not go back. So we want to combine standardized clinical outcome measures. So that means every clinicians measure the same thing in the same condition with the same material in clinical sites for all the patients. So it means that we need to agree on the way to do it, what to measure. So that's a first thing. Then we want to integrate the remote real world data using wearable technologies to those first data. And really the goal is to capture the disease progression. And so we are going to quantify mobility, activity, sleep, all the cardiac, respiratory, different physiological parameters in daily life. On the other hand, we are developing in collaboration with Combined Brain a disease model based on patient experience. Maybe some of you are participating in that study. So you can participate if you are from Europe and you speak English, and US or Europe, sorry, and you speak English. I asked for Israel and Australia, and they didn't include it in the IRB, so in the authorization from the ethic committee. So if you are in Europe and you speak English, you can participate to that study, and you need to hurry up because we really need patients, and it's going to end in a few months. So basically, you answer a survey, and then you will be, so they will check if you are eligible for this kind of study, and then they will contact you and have a conversation with you. So they let you explain the way you live Myhre syndrome. It's not
Amel,
questions.
Amel, you said that this survey is not open for Israelis, but if our daughter has also American passport?
That's a good question.
I can check with Mary. That's
Yeah,
a really good question. Yeah, I'll
Okay,
check for
thank you.
you.
Thank you.
yeah, that's yeah. Or if you have an address in the US, I don't know.
No, but let's see. Let's see.
Yeah,
She will tell. Mhm.
yeah, that would be really helpful. Um, so I just put why does it matter to have a good clinical trial design, but I think everybody knows why it's matter to have a quality in the clinical trial. But I think where I would like to insist is that if you choose the wrong endpoints, so the wrong things to measure in patients, you can fail. A good compounds, a good treatment can fail if you don't measure the right things. And it happens so many times in the past that you have a good drug, you give it to the patients really feel better, have a better quality of life, improve, but do not improve in the single thing that the clinician said they are going to measure. And then they go to the FDA. The FDA says, well, you didn't improve this QT on the cardiac thing, and so it's not working. And all the patients say, yeah, it's working. I can move, I can uh, go to the shop, it's working. And they say, no, we cannot accept it because you, in fact, originally you didn't measure the right things. So now the FDA is more and more open to the wearables technology and the real world data because they realize that that's the best way to see if a drug is working for the patient. So that's the reason why we really want to include that in the clinical trials. So what the thing we do is to try to align, as you were saying, globally, all the clinicians everywhere. We are working on the clinical care guidelines. So we gathered 41 Myhre syndrome clinicians from Australia, everywhere in the world, all the people that we know. So if you know other clinicians that have, the criteria was to have at least 3 patients or have 2 patients, but with a very special complication. That can be useful as information also. But we have 41 clinicians that see at least 3 patients or more. We gather them. We are doing a Delphi study with 139 statements. So it's a lot of work. And they are scoring, and we are trying to get a consensus on those 139 advice on how to care for the patient. We did the round ones, and I'm analyzing the data, and then we'll start the round 2 in about 3 weeks. So what's good is that we see that 41 clinicians, I asked, I don't know, 43 clinicians, and 41 in less than a week said, yes, I come here, I will spend some time with you, and I want to participate to that study. So that's it shows that Myhre syndrome clinicians are really involved and committed, and they want to do well for the patients. On the other hand, the clinical trial design working group is a smaller group, but maybe with the occasion we had to gather everyone, 41 clinicians for the clinical care guideline, I'm going to increase the number of people in the working group to get a consensus on the outcome measures. So what to measure in the patients and how to design the clinical trial. So the next step is in autumn 2026, we are going to organize an in-person expert meeting with the clinicians to finalize the clinical care guidelines, get really a consensus on how we are going to write the guidelines and get a consensus on the outcome measures and endpoints. And that will be in Paris. So that's what we did mainly. That's the main road we had and what we are going to do. But aside from that, we had side projects that are necessary to go ahead faster. So we want to obtain an ICD-10 code for the US patients. We are expanding the biobanks in Asia and in Europe, where you will be able to send the blood samples from the kids or the adult patients. I think, Matan, you sent already, the clinician sent already the blood samples to Belgium, to the biobank in Europe?
I send you an email that we need. We found just now a lab that can do it. So we see in my
Okay.
email. Okay.
Oh, okay. So I'll get in touch with you to
Okay.
get better. To go ahead. Sorry.
Mhm.
So we are working on engaging pharmaceutical partners because we cannot do that, get the drugs and pay millions of clinical trial by ourselves. We collaborate with a line foundation that have the same targets as we have and are going after the same drugs. So instead of competing, if we get together to go to the companies and try to get the drug in our patients, I think it will be more efficient. We strengthen our non-animal model platform recognition. That's important because people realize more and more, and people from the FDA, from every kind of stakeholder realize that starting from mice is not a good idea. And it's more efficient to start from non-animal models. So our platform is relatively innovative, and we are positioning ourselves as innovative people with now experience in non-animal model. We want also to develop innovative delivery systems, so especially for the gene editing, but also for small molecules that are efficient but do not penetrate the brain. So we are exploring with Australian. We're just starting to talk with them, so it's nothing yet really going on. But that's the first step, was to see how we can develop something efficient to get our treatments everywhere in the body and not only in the liver or special organ. So that's oops, that's it. I will stop sharing.